Okay. Another reminder that you have to take first midterm this weekend, somewhere between Friday noon and before Sunday midnight to be finished. It's on D2L, 90 minutes to take it, 100 multiple choice questions on chapters 1, 2, 3, or the prologue. Questions about the course or the test? Good. Yeah. Amy. And all of chapter 3 will be there. We will finish talking about it today. Hopefully in time to have a Kahoot, or maybe even 2. So we'll see. All good? Okay. We were talking about the neural crest. That is that line of cells when the neural tube forms and the ectoderm closes overhead, and now you have this disembodied neural tube running the rostrocaudal length of the animal's body. And beside it are 2 rostrocaudal columns of neural crest cells, also kind of separated from the rest. And because they're being exposed to different amounts of factors from the floor plate factors, sonic hedgehog, and from the roof plate factors like bone morphogenetic protein, things like that, because they're getting different signals controlling what sort of gene expression they're having, they already have distinct fates. And it's going to be the neural crest cells. They're going to start shinning around the body to take on and form the peripheral nervous system. And some neural crest cells go to become and invade all of the skin to make the pigment epithelia, melanocytes, so-called. Others migrate a very short distance and start forming dorsal root ganglion neurons that send their unipolar axons in 2 directions, into the spinal cord and out to the body. Some take up, a very few take up a position in the midline, not too far from the notochord, where they will take on the role of autonomic ganglia, and even fewer yet will migrate to the middle of the notochord, where they're going to be taking on the fate of the adrenal medulla. All neural crest cells. We'll talk about them some more later, but the neural crest cells, they're also going to provide the myelination of the peripheral nervous system in the periphery. And what are the cells that myelinate axons in the peripheral nervous system? Schwann cells, right? Not oligodendrocytes. Those are a different cell lineage, and those are only in the central nervous system. So far, so good. And lying beside this tube of the neural tube, there are 2 smaller tubes lying parallel to it, one to the left, one to the right. And these cells are mesodermal in origin, and they are going to form somites. Eventually, as we'll see, they'll start expressing different genes in different parts of that column, and they will eventually coalesce into these blocks, side by side, somites, a block of somites on the left and the right. And not so coincidentally, those somites, there'll be exactly as many of those as there are going to be vertebral segments in the spinal cord. So they're all previews of that. And those mesodermal cells, they're going to make muscles and other stuff that are going to be in that portion of the body. Okay? And as I told you, the fate of these neural crest cells, which of these roles they take on, depends on where they are in the neuraxis, in the rostrocaudal. So, for example, some neural crest cells in this area that's in yellow, they're going to migrate out and start shinning up and down the primitive gut, and they will eventually form the enteric nervous system. That nervous system that's keeping things working properly for digestion and that you have no control over. And whether or not you consider that part of the autonomic nervous system, I do, because you don't have control of it. But more traditionally, the autonomic nervous system, people think of it as being the sympathetic nervous system and the parasympathetic nervous system. And I'll remind you, the sympathetic nervous system, it was originally called the thoracolumbar nervous system because it only originates from the thoracic segments and the lumbar segment. right? So that can help you to remember that. And those neural crest cells are going to migrate along the vertebral column, one on either side, and they're going to start breaking into lumps. They're going to form the sympathetic ganglia, also sometimes known as the sympathetic chain, because there is this chain of ganglia that are on either side of the vertebral column that are going to receive input from the spinal cord, and then the cells that are in these ganglia, they're going to send their axons out to periphery to provide sympathetic nerve innervation. All the fight and flight stuff is going to be controlled by these neural crest-derived cells in the sympathetic ganglia. And they'll do things like make your iris get bigger, speed up heart rate, speed up respiration, prepare glucose mobilization for energy, etc. And those neurons in the sympathetic ganglia, they're providing sympathetic innervation to the body. What neurotransmitter do they use? They're speeding things up. So if you're British, you could say they use, Nope. Yeah, so if you're British, you'd say it's noradrenaline, right? You know, adrenaline speeds things up, but Americans call it norepinephrine, right? So they're going to use norepinephrine for a transmitter. But indeed, another subset of the neural crest cells are going to become autonomic ganglia out there in the periphery, and I'm not showing them on this chart, but they are going to use acetylcholine to do the opposite, generally, of what the sympathetic nervous system does. Slow down the heart, as opposed to speeding it up. Stuff like that. You okay? So there's a very close relationship between where they are in the rostral caudal axis and which role they're going to take on. And how do they know where they are in the rostral caudal axis? Well, because of those cues we talked about last time, right? Things like the one that's easy to remember is how retinoic acid, right, is more concentrated in the caudal end, etc. But once they start migrating, they're going to take new cues from their pathway that are going to direct them to take on the fate that's proper for where they happen to be. So in order to understand how they do that, we have to talk about migration in a later animal. So far, when we talked about cells migrating around, there hasn't been much for the cells to migrate around except each other. But what's happening once development proceeds is cells start secreting these long chain molecules into the environment around them. So each cell is making its own little coating of stuff. And we call this stuff around it the basal lamina. Lamina as in a layer, and basal as it's right next to the membrane. And it's the cells themselves that make this basal lamina. And you've heard of some of the molecules that are involved in this: collagen, laminin, fibronectin, integrin, intactin, the heparin sulfate proteoglycans, things like that. What they all have in common is they're really long chain molecules, and they love to stick together, and they love to stick to things. So they're sticky. And you can think of them as providing structural support for the shape of the cell that's forming there. Some parts of the body have really thick layers of this stuff. And if it's a really thick layer, we may call it an extracellular matrix. And when we do talk about biology and, stuff like that, we pretend that this extracellular matrix, the basal lamina, we pretend it doesn't exist. But in fact, it's a fact of life. And so all the things, all the ways cells are communicating with each other, they always have to deal with the basal lamina. And by the time the neural crest cells start migrating, there's enough basal lamina out there that the first thing, that they're all being hemmed in. And so the first thing they have to do is start secreting stuff to dissolve the basal lamina so they can start moving. Got that? So earlier they weren't expressing the cells, but then they start expressing metalloproteases. I don't need you to know which ones. They're called proteases because they dissolve proteins, right? And they're called metalloproteases because all of them require a heavy metal as one of the ingredients to hold the enzyme together. Okay. And so they start expressing and secreting the metalloproteases, and you can think of them, they're loosening themselves up, and now they can start migrating. So far, so good. And once they start migrating, it turns out that column of somites on the side, they are not homogeneous for the entire rostrocaudal length. In fact, various clumps of somites start expressing stuff called ephrins. Remember, ephrin is a signaling system where one cell can release ephrin into the environment, and other cells have ephrin receptors that respond to that. And often, ephrins serve as repellents. It's not that they attract cells to them, they push cells away. And so what's going to happen is the ephrins are being expressed in the somites in the posterior half of each somite. And at this stage, a somite is a length of that column that's going to, eventually give rise to a spinal segment. And it turns out neural crest cells really hate ephrins. So if you look in this photomicrograph where they labeled the neural crest cells with green fluorescent protein so you can see them, and then they also labeled in the somites, which was a posterior half because it was labeled for ephrin. And you notice that the neural crest cells refuse to migrate over ephrin. This then is the beginning of spinal segment, right? These neural crest cells, remember, some of them are going to make dorsal root ganglion cells, right? So the reason there isn't a continuous line of of spinal roots coming out, a sheet of spinal roots, they're not. They come out in segments. And this is how they get divided up into those segments. And that's good because eventually the spinal cord is going to have a vertebral column made around it, a bony vertebral column. And as you know, the spinal nerves have to go out through little tunnels between one vertebra and the next one, right? That's how they get out. And so this is why the spinal cord is divided functionally into spinal segments, because the neural crest cells have to avoid the ephrins that are being expressed in the posterior half of each somite. And although they avoid ephrins, there's another stuff called fibronectin and another thing called neural cell adhesion molecule, or NCAM, and neural crest cells love to migrate on that. They love to touch that and move along it. And here's an experiment where the positive lanes are coated with Ephrin, and you can see again, as the cells that start off down here, as they're migrating this direction, they're avoiding the lanes of ephrins and coming out in discrete lines. And what is this dish coated with? Fibronectin. So they really like to attach to fibronectin. And what's fibronectin? Oh, remember, that's one of the things that can be found in basal lamina of other cells. Yeah, good.
Clinical Ephrin station, cells repel from Ephrin. So what cells have the receptors that bind to the Ephrin?
All right, I see. Great. It is the neural crest cells themselves that have the Ephrin receptors. And so they're avoiding Ephrin. And in general, Ephrin plays a repulsive role. We'll see that again when we look at the development of the optic system, where again, cells with lots of Ephrin receptor don't want to be around Ephrins and vice versa. Yeah.
So this cell has more Ephrin, is that how we get different segments, so like the rhomboid and then the sacral or the lumbar? How?
No, that specification of sacral, lumbar, etc., that's something else. I mean, but they all have segments, and they all have somites, and the posterior half of all the somites are expressing the Ephrins. Laura?
So what's the point of having Ephrin receptors if you're going to try to get away something?
Yeah, what's the point of having Ephrins there if it's a repulsive cue? And it's a great question, but I'll point out to you that, repelling things can work just as well as not, right? So just as well as guidance. So imagine you're on a highway and there's an arrow that says go this way, okay? Or I can also direct traffic by putting barrels in one lane, and by golly, you're going to go that way, right? Repulsive factors can be provide just as much guidance as attractive, and we'll see that over and over again. Yeah. But it's a good question, because I can tell you that as people started finding these adhesive molecules, again, I'm old enough, I remember when these were all new things, that came as something of a surprise.
Mm-hmm.
And when we talk about growth cones later, we'll find what surprised for people was a growth cone, which is the part of the axon that's reaching out. You can watch them touch a repulsive factor and the growth cone just collapses. Like, oh no, that's a no-go zone. I'm not going that direction. And that was a surprise at the time. But then you step back and you think, well, repulsion works just as well as attraction. Nicole.
And
Erica, sorry.
So does a concentration like Ephrin determine like the second?
Does concentration of Ephrins determine?
Like the second?
Not at this stage. Later, we'll see gradients of Ephrins, but that's not what's happening here. This is pretty much off or on. Yeah, Angelica?
So is collagen and the fibronectin also considered cell adhesion molecule?
No. I mean, they can serve as that purpose, but there are some cells that got discovered as cell adhesive factors, and so we'll try to restrict it to NCAM, and then later we'll run into NCadherins, et cetera. So I just try to keep the terminology separate. And in terms of whether it's an adhesive molecule or not, I mean, to some extent, it's just semantic. But they're all sticky. And cells are paying a lot of attention to where they are and where they're not. And that same signal of having the Ephrins in the posterior half, shortly after the neural crest cells start migrating, the first neurons in the spinal cord will arise, and they are motor neurons, and they send out their axons very early in development, long before there's a body out there, and the muscles are just barely forming. And they too avoid Ephrins. And so this is why the neural crest cells, because they avoid Ephrins, that's why the dorsal root ganglia, the dorsal nerve and the dorsal root ganglia are in particular position, and the ventral root from the motor neurons is going to also be in that same rostrocaudal position, the ventral root for this particular portion of the spinal cord. So that same cue is also going to make sure the ventral roots go out in that opening that eventually the vertebrae are going to leave open when they start forming, because there are no bones yet, a long way off. And I told you that the fate of neural crest cells depends on various things they get exposed to along the way. And one piece of evidence of that is it turns out neural crest progenitors, unlike neural cells, are relatively easy to grow in a dish. You just got to make sure you provide the right sort of factors there for them. And we'll talk about many of these things later. But once you have these neural crest progenitors growing in a dish, you can expose them to different factors. Don't worry about what the names are. And that can direct them to start differentiating into a particular neural crest fate. And in some cases, it's not just a factor, it's got to be a sequence of factors. First, one of the FGFs, and then NGF, and they start looking adrenergic. If it's first an FGF and then something called CNTF, then they start looking like cholinergic neurons. And so the fact that you can direct their fate so readily in a dish tells you that they are pluripotent and that they're listening for cues in the environment to take on the normal role. And we'll talk about some of these factors and some of the pathways again in future chapters. So this is a preview in that regard. And I'm not going to go over this in detail. As I've already told you, there's a lot of different fates for neural crest cells. Do I have this in two parts? Yeah. Of neural crest cells. And I think I've talked about most of them. They're going to form the cartilage and some of the bones in the face. And what else? What have I not maybe talked about? We'll never talk about the carotid body again, trust me. Oh, and some parts of teeth, etc., connective glands, etc. So they're playing an important role in making a body. And the peripheral nervous system. And I'll take a little bit of time to point out to you this table about the various fates of those neural crest cells in the pharyngeal arches. So, and to be clear, it's not that neural crest cells alone make the pharyngeal arches. Other cells contribute too, but neural crest cells are part of that mix. And depending on which of the pharyngeal arches you're looking at, they'll be making different things. So in this part, the neural crest cells will start making those ganglia for some cranial nerve nuclei, the trigeminal, some of the trigeminal nuclei. And you remember why the trigeminal nuclei are called that? Because there's three branches, right? One for the face, one for the upper jaw, and one for the lower jaw. So your dentist, when he's numbing one of those things, he's putting lidocaine on one of those three big branches, never the facial, obviously. Or never on purpose, right? Anyway, others of them are going to make those, some of the middle ear bones. Others of them are going to make bones in the throat, and etc. And various branches of the cranial nerve nuclei. And I'll remind you, the cranial nerve nuclei are clumps of neurons that are not central nervous system, they're peripheral nervous system derived, and it's the neural crests that populate those. They'll send one axon into the brainstem and another to the body, or they may receive an axon from the brainstem and then send an axon to the body, depending on whether they're sending a sensory information into the brain or sending motor commands out of the brain for you to do things like eat a blow pop and, crunch it at the end to get your gum. Questions about that? No, I don't want you to memorize which pharyngeal arch gives rise to which, right? But I do want you to know that they make the otoliths, the three middle ear bones, and stuff like that. I want you to have an idea of what they contribute, but there's no point in memorizing which does what. So that's all I'm going to say about neural crest and migration right now. It's another whole topic coming up in a future chapter. Okay, we'll talk in this in more detail. And you'll learn why what used to be called leprosy, Hansen's disease, it's neural crest cells play a big role in that. Really? Yeah.
Awesome.
Yeah. No, it's good. You should. Every once in a while, you should say that. You should just say, "You're kidding," or something like that. And it's, yeah. Yep. See, and now I want to tell the story now, but it's next chapter. Okay, I'm going to switch topics. We're going to talk about the cerebellum. We've talked about the cerebellum some already, and here's a section through the cerebellum that's been color-coded, doesn't matter what in particular. But I'll remind you that the cerebellum also has a cortex. All that means is the outermost portion. And what defines the cerebellar cortex is this row of massive neurons called Purkinje cells, each of which has in adulthood, has an enormous dendritic array. And so, how big are Purkinje cells? If you make them dark enough and give them a little contrast, you'll see them with your naked eye. The dendrites aren't that big, but as dendrites go, they're really big too. And each Purkinje cell, you can think of it as being like your hand, where the palm represents the cell body, the fingers sticking straight up represent the dendrites, and your wrist represents an axon that's going to go down to eventually innervate the deep cerebellar nuclei. Don't need to know that, just reminding you. And that each Purkinje cell's dendritic array is two-dimensional. It's only in those two dimensions, like the fingers of your hand. And if you look at any two neighboring Purkinje cells, their dendrites are lined up in the same plane, parallel to one another, on across fold after fold of cerebellar cortex. Abner.
Why are they like that?
Yeah, most neurons, right, when you see them, they have their dendrites sticking in lots of different directions. No, there's a reason why the cerebellum and the Purkinje cells are so well lined up. And we'll talk a little bit about that. What I won't tell you is why they're two-dimensional, because I don't know that everyone knows that. But I will tell you how they get to be the size they are. And as we talk about the development of the cerebellum, we're going to learn something about the circuitry of it, and you'll see that anyone looking at it would know this is a timing device. Any human engineer who built things wired this way would be doing it in order to keep track of time as an axon, because think of those hands with their fingers up and they're all parallel. Pretty soon we're going to see that there are fibers that go across those dendrites, and the same fiber will provide synaptic stimulation first to this Purkinje cell, then its neighbor, then its neighbor, then its neighbor. And since action potentials take time, there's a very set amount of time between these stimulations. If you understand how it can be a timing device from my having said that, you're a genius. So don't think you should understand. I'm just giving you a hint. You okay? So far, so good. And in the cerebellum, we'll meet another kind of glial cell, the Bergmann glia, and we'll see that they play a role pretty similar to radioglia. They look a little different than radioglia, and so they have a different name because a guy named Bergmann found them first, right? But they play the same role. And the development of the cerebellum is unbelievably interesting. So in the area of the pons, the neural tube, it's been growing while the telencephalon is growing, et cetera, right? The metencephalon back here has been growing. And of course, there's a tube in the middle of it, which was the tube of the neural tube beginning, right? But as things grow, the tube gets more complicated in shape, and it's going to make what we call the fourth ventricle. And what begins to happen is that there's a portion of the ventral half that has been given the name the rhombic lip. Doesn't matter why. And what happens is there are a bunch of cells there that start migrating up over the surface of the dorsal half of the neural tube here. So it's like they're getting out of the car and crawling over the top of it. And these are granule cells, meaning they're really little. And so for both the left and the right, they migrate from the rhombic lip onto the top of the developing Purkinje cell and form the external granule cell layer. And they are dividing while they're migrating. And when they get out here, they continue to migrate. So, they're dividing while they migrate. And as they come out here, they continue to divide. So they're dividing like mad. And pretty soon you've got this big population of granule cell neurons to be out there on top of this part of the neural tube. And in the meantime, in the ventricular zone of that dorsal part of the neural tube, there are cells that have been giving rise to Purkinje cells. And the Purkinje cells are all lined up, and I'll tell you how they know where to be in a moment. They all line up so that they form one layer Purkinje cells. And their dendrites start growing up. And at the same time that they're getting here, they start secreting sonic hedgehog. Yep, the same old sonic hedgehog. But for the cells in the external granule cell layer, for them, sonic hedgehog induces them to divide some more. So it serves as a mitogen, a factor that encourages mitosis. How you doing so far? And in the adult, there will be no granule cells on the outside of the cerebellar cortex. What happens is these granule cells eventually start migrating down. Guess what? They're holding on to Bergmann glia. They start migrating down. And as they migrate down, they send out dendrites. And as it keeps growing down, the dendrites keep growing out. And I couldn't show it this way in this, but these dendrites are now going to grow perpendicular to all the Purkinje cell dendrites that are growing. I didn't mean to say dendrite. These neurites, you call it a neurite when you don't know if it's going to be a dendrite or an axon. These neurites are growing perpendicular to the Purkinje cell dendrites, and eventually are going to be axons. They're going to synapse on each of those Purkinje cell dendrites. Because they're massive in the adult, and they're all sending out these axons perpendicular to the plane of the Purkinje cell dendrites, they're known as the parallel fibers.
Okay.
Cahal called them that. Very striking, because they're all parallel to one another. And the cell body keeps migrating down and will eventually lodge itself in what we're now going to call the internal granule cell layer. Here's another view of this. So here are the Purkinje cells. Here's the external granule cell layer. And here we're taking one particular external granule cell that starts to differentiate. It's now committed to becoming a neuron. It sends out these dendrites that are now, maybe you can see that they're perpendicular to the two-dimensional array of the Purkinje cell dendrites. And they keep going down, down, down. And eventually, they're going to be down here. And they're going to interact with other cells. They're going to get stimulated themselves. And their axons are going to go up and travel and hit one Purkinje cell dendrite after another, or the dendrites of one Purkinje cell after another after another. Very highly organized structure. And again, it was Cahal who worked out this structure, and he was blown away at how predictable it was, right? How regular it was. And so there's some other players here that we aren't going to talk a lot about. It'll turn out that from the ventral heart part of this metacephalon, there are cells that are going to form the so-called inferior olive, and they will send their axons up, and their axons love to glom on to Purkinje cell dendrites. Other cells are going to come up and form synapses on those granule cells, and because the synaptic array there looked kind of mossy, Cahal called them mossy fiber terminals. What they do and how they affect Purkinje cells, we don't care. We're just trying to see how all this happened during development, during ontogeny. Okay? Early in the 20th century, in the late 19th century, Americans became fascinated with having mice as pets and breeding mice as pets. And they picked this up from East Asia because China and Japan had been breeding mice and selecting them for domestication for at least for hundreds of years, maybe for 1,000 years. And what the breeders in Japan and China had done over the years, they had isolated some strains of mice that they thought looked funny in their behavior. Americans called them dancing mice because they never went anywhere in a straight line, right? They were reeling around, dancing, moving around. I'm not sure it was fun to be a dancing mouse, but for the fanciers, this was interesting that, they were different and you could breed them. So it was clearly a genetic trait. And what all those dancing mice have in common was misshapen cerebellums. And eventually, once the genes were isolated, what all those mutations had in common is that they were things that disrupted the migration pattern I just told you about. Now, there are probably some other mutations that disrupted it so much those animals died, right? But these were the ones that over the century had survived long enough. And even though they were pretty disabled in terms of motor behavior, they could still copulate. And so that's why we still have some. If there were some that so severely motor disabled that they couldn't even copulate, then, they didn't make the cut. So here are some examples of some of these cerebellums. Once the genes started getting isolated, they got named various things like Weaver and Reeler, etc. And so here's what a normal mouse cerebellum looks like. Here's what they look like in Weaver. Shrunken, and if you look, you can see the Purkinje cells still in a row, but where are the granule cells? Oops, they're gone, aren't any? And so in this animal, the cerebellum is basically non-functional, and the fact that it can get around at all is probably due to plasticity. But the help that the cerebellum normally would have been giving it to get around is basically disabled. And Reeler is even more severely deformed. If you look here, the Purkinje cells, instead of being in a row, they're all over the place. And the granule cells, well, at least they have granule cells, but instead of being in the internal granule cell, they're still all out on the outside. Totally dysfunctional cerebellum in these mutants. To try to understand the nature, the mechanism behind, I control this story yet. That business of it becoming a hobby to breed mice, it really caught on, especially in the East Coast of America. And there was a funny thing that started happening, which was that in those days, school teachers were not allowed, they were all women, and they weren't allowed to marry. If you got married, you lost your job because, my God, you might show up pregnant sometime. Children must not know that this happened. And so there was a whole cadre of women who, when they retired, had no family to devote themselves to. And so many of them took up breeding mice as a hobby. And all these inbred strains of mice that some of you study in the laboratory, how do you make an inbred strain? What's an inbred strain? An inbred strain of mice are those in which 99, for each animal, they have the same alleles at 99% of the sites on the genome. It's easy to make such animals. All you have to do is do brother-sister mating for at least 10 generations. But the problem with that is when you're doing brother-sister mating, after a few generations, you get hardly any offspring because eventually you accumulate recessive traits that are fatal, lethal, right? But, okay, you start 100 different brother-sister mating lines and keep going down, and many of them peter out. But if you can get to the point where you've got to the 10th or 11th generation of brother-sister mating, now these animals are homozygous at every site in the genome. And now you can breed them to one another. And so the C57, how many of you ever studied C57 black 6 mice? or bald mice. All right, you can thank those retired school marms because they were the ones that went to all that trouble, and a guy at Harvard helped, coordinate with them, etc. Okay, back to dancing mice. What's going on in these animals? One way to get at that was to try to simplify matters, and people found that they could take, if they took embryonic cerebellum, they could isolate granule cells and Bergmann glial cells and get them to grow in a dish. And one of the interesting things they found is that if they isolated a whole bunch of glial cells and put them in a dish, they would just sit there like lumps. Only if they added granule cells to the dish, now the glial cells started making these long strands, just like in the animal. And not only that, the granule cells that they put in there started migrating on. They'd get on one of these glial cells and migrate all the way to the end, and then turn around and migrate back again. Two things that's being shown. Number one, again, cell-cell communication. The radioglia don't form those processes unless some neurons are around. So somehow the neurons induce them to take that shape. And once they do, the two recognize each other, and the granule cells, they're ready to shinny, right? They're ready to shinny up there. And if they, yeah. So people use this to try to get at the defect in Weaver. And so they reason like this. Is the problem with the Weaver mutants, is it a problem in the radioglia, or is it a problem in the neurons? One of the things they noticed is that when they put them in the dish, wild-type granule cells would go up and down, but when they made these things from Weaver animals, the granule cells just sat there. And so to get at what's the problem, is it the Weaver glial cell that's defective or the Weaver granule cell that's defective? They did situations where they got glial cells from one animal and granule cells from another, and you could already know what they're going to do. What happens if you put together wild-type granule cells and put them on into a dish that has a bunch of Weaver glia in it? Everyone get that? And lo and behold, they migrated just fine, happily as could be, back and forth. It was like they were from normal animals. But when they did the converse, this is here, but when they did the converse, where they had Weaver granule cells and wild-type glia, no migration happened. So they knew that whatever Weaver was doing, the mutation was doing to disrupt migration, it was causing the granule cells not to behave properly. And this is an example of a cell-autonomous effect. That is, the effect of the Weaver mutation was acting in individual granule cells. So it's the granule cell in which the Weaver mutation was having its effect, not the glial cell. So far, so good? Okay. And the problem with reelin turned out to be that reelin was a secreted protein that is normally produced by the granule cells. And the Purkinje cells, as they're migrating up, they're paying attention to the gradient of reelin secreted by the granule cells. And they stop when they reach a certain concentration of reelin. And since the granule cells are putting it out equally, that ends up with a single layer of Purkinje cell bodies, each of them lining up to be just under the ceiling of the reelin protein. And the mutation in reelin disables that cue. And so the granule cells don't secrete reelin. The Purkinje cells don't know where to go, and they end up being all over the place. And because they're all over the place, the external granule cells never get their cue to start migrating, and they just stay out there in the wrong place. And so that's how you end up with the granule cells in the outside and the Purkinje cells everywhere, and basically you have a non-functional cerebellum. One more migration story to tell you. You're ready? And that involves Kallmann syndrome that you talk about in the book. There are certain places where non-neural cells start invading the developing nervous system and take on a function. And one of these examples is that there are some cells that are out in what's called the olfactory plateau. Still ectodermal derived, but it hasn't been going down the neural pathway. And then And so it turns out that there's some cells in the olfactory plateau that start migrating and migrate along olfactory sensory neurons. So the olfactory sensory neurons are there already. Remember, they're going to send their axons up through the cribriform plate into the olfactory bulb and synapse there. We're not showing those synapses because that's not the point here. But these cells invade the brain by climbing up the olfactory sensory neuron axons and into the brain, and then they start migrating and they end up in the hypothalamus. These cells, even as soon as they start migrating, they're already expressing an important signaling protein in the endocrine system, gonadotropin-releasing hormone. So they are already distinct because they're making GnRH. And that's good because we can use immunocytochemistry to identify them, and we can watch them as development proceeds, go further and further into the brain. And then they settle in the hypothalamus. They send their axons down to the median eminence, and they're going to release GnRH into the hypothalamic pituitary portal system and cause anterior cells there to release gonadotropin, follicle-stimulating hormone, and luteinizing hormone. We'll talk about all these hormones more in chapter 7.5. But for now, because we're talking about migration, this is how they get there. And some of these cells are in fact neural crest derived, but not all. Yeah.
Yes, it.
Pardon?
Reason they want to invade.
Is there a reason they want to invade? This is the sort of thing that happens in development that no human would ever arrange, right? This is just not, why go to the trouble? But natural selection doesn't care what you find reason, right? In the common ancestors of vertebrates, this is what happened, and this is how regulation of gonadal function came to be. And I guess it works because look at all these vertebrates, right? So no, no one's ever come up with a reason why it had to be that way versus some other way. That's just how it was. Probably in some common ancestor we shared with lampreys. Not very satisfying answer. You okay? And so these cells are going to be crucial for the animal to later become reproductively competent, because it'll be the stimulation of the gonadotropins from the anterior pituitary that will cause the gonads to start making gametes and start secreting hormones so this individual is going to reproduce. Here's a view of that. So we're talking now about the GnRH cells. They've migrated in. They've settled in the hypothalamus. They send their axons to the median eminence. They release GnRH into the hypothalamic pituitary portal system, where it goes a short distance and acts on anterior pituitary cells. And they, in turn, release gonadotropins, either follicle-stimulating hormone or luteinizing hormone. And although they're named for what they do to ovaries, they are also crucial for male reproduction. They're also going to be crucial for pushing testicular development. Occasionally, there's a mutation that disrupts this migration of GnRH cells, and that by itself is not lethal. But if that happens, then you can get something known as Kallmann syndrome, where these are people who the age of puberty comes and their gonads never start working. And that's because there's no GnRH to start the anterior pituitary to release gonadotropins, and so the gonads don't develop. And so if women, menstruation doesn't come, and they go to a doctor to find out why, and eventually this will get diagnosed. Or in men, again, puberty never comes, no armpit hair, no pubic hair, and in both sexes, they tend to get kind of tall because body growth is mostly halted by gonadal steroids that stop long bone growth. And so they can be kind of tall. And in many cases, what they have in common is not only do they not undergo puberty, but they also can't smell. Because in some cases, the problem is that the olfactory system doesn't develop properly, and that's why they're not going to be able to smell. And without those olfactory sensory neuron axons for the GnRH cells to migrate along, they never get into the brain. And so this is the case. Another example, and these same genes are also important for ear development. And so in many of these cases, they may be partially deaf or even entirely deaf. So pleiotropy at work, 3 different symptoms, all caused by the same mutation. And they seem totally disparate and unrelated. Smell, puberty, hearing. What do they got to do with each other? Nothing as functions, but the underlying developmental mechanisms behind them happen to use the same gene. And that's a classic pleiotropic effect.
So they miss puberty phenotypically. Do the kids look female and male?
They look male and female, and that's an interesting question too. Were you in human sexuality? No. Okay, you take it next year. It turns out that their sexual differentiation in the first trimester of pregnancy is perfectly normal because early fetuses don't make GnRH. Nothing to do with estrogen. Early fetuses don't make GnRH, but the placenta does. And so gonadal development in the early stages of fetal development, that's being driven not by any GnRH that the fetus itself is making. That's being generated by moms providing gonadotropin from the placenta, right? Human chorionic gonadotropin, right? It's going to cause the gonads to develop. They'll be secreting testosterone in the males, and they will cause sexual differentiation in the periphery. So they look like typical normal males, but they never undergo puberty. Typical treatment is to provide them with the steroids that they need for one sex or the other for the rest of their lives. Yeah, Addie.
So what people with the syndrome have like a smaller anterior pituitary?
That's a great question. I don't know. Do they have a smaller anterior pituitary? They might, but remember that some of the cells in the anterior pituitary have nothing to do with gonadotropins, right? So others of them are making TSH and CRH. Yeah, so I don't know. Yeah, and okay.
You need someone to have puberty even past like this, right?
Yes. So if you provide them with gonadal steroids, estrogen and progesterone for women, testosterone for men, then they'll undergo puberty at that point. And, with mixed success. The individuals are never going to be able to reproduce if you do that, right? Because there's no gonadotropins to cause gametogenesis. And so, but otherwise they can be like other folks. There is some effort underway to treat them not by giving men testosterone, but let's give them GnRH. Or let's give them gonadotropins, GnRH went up. And in that case, the gonadotropins might cause testicular development enough for them to make gametes and stuff like that. And I don't know where that stands. Laura?
You can fix that by giving them hormones.
Yeah, great question. No, you can't repair the hearing with hormones. If it's total deafness, of course, those are excellent candidates for cochlear implants and things like that, or they're deaf. So nothing wrong with that.
Hormonal signaling pathways like effective, or really just?
As far as other hormonal signaling, you mean like growth hormone or thyroid hormone? Not that I know of.
Next.
Yeah, so because it's only these rogue cells. The other thing I'll tell you about GnRH cells is something I always thought is kind of strange. Most of the time when you talk about a functional group of cells in the brain, you notice how they're close to each other, right? The GnRH cells, they don't make a nucleus. They're just kind of spread out in the hypothalamus. You have no way of predicting from one individual to another where they will be exactly. And there's only a couple of hundred of them. I mean, absolutely crucial for reproduction. But it's like they don't need to be neighbors with each other. It must be that the synapses they get to decide when to reproduce, it must be that that's being quite selective. But yeah.
There's a testing identify common syndrome prior to puberty.
Typically it's not. It isn't. I mean, it isn't usually identified before puberty. No, because I mean, in some cases they can't detect odors, but people don't notice, and their families don't notice, right? So, and the earlier it's identified, the better, because as in the vignette in the book, the gentleman felt like it was so late. First of all, he was totally embarrassed that he's 22 years old and a biomedical researcher, and he still hasn't got any pubic hair, underarm hair, and he still looks like a boy, just really tall. And in his case, testosterone it was a little late for it was only partially masculinizing. And so he felt like that was really bad. And when he went to doctors about the late puberty, almost nobody thought of this syndrome. They just, well, you're a late bloomer. And he got to be a later and later bloomer. And it's not till he hears a guy giving a talk that he goes, a scientist giving a talk at this English research center where he worked. And he went to talk to him afterwards and told him, and the guy said, well, wait a minute, can you smell odors? I said, well, no one ever asked me that before. Oh, I might know what's going on here. Okay. And you can, if you want to, you can look up those things but I want one more topic. Okay. This is the cutting edge of this chapter. This is going to be a challenge for both of us. Okay. I'm warning you that ahead of time. In modern talks, you're going to see lots of things that look like this, which are plots that are plots of patterns of gene expression in different kinds of cells. Okay? And I want to try to explain to you how these plots get made. And to do that, a little terminology. These days, you can sequence all of the mRNAs that are being made by a single cell. So let's pretend there are 20,000 genes in a mouse, somewhere around there. You can take a single cell out, and the techniques are powerful enough now that you can get a read of the relative amounts of every mRNA that was in there, which is to say, it's giving you a read of the pattern gene expression that particular cell had before you killed it and took out all the mRNA. So far, so good. And it's quantified. A whole bunch of this, a whole bunch of that. Nothing of many genes, right? But a particular pattern. And this business is known as RNA-Seq. And you can produce then a transcriptome of that particular cell. What are all the transcripts that this particular cell happen to have? And there'll be a whole bunch of one, a little of another, and none of many other genes. And if you think about it in terms of cell differentiation, tell me the pattern of gene expression that cell has, and I'll be able to tell you, if I knew enough, what particular function it's going to serve, and even what particular structure it'll have, right? Because that's its function. So you can repeat this for thousands of cells from the same animal. The amount of work involved is monumental. But happily, much of it's automated in terms of the RNA-Seq and stuff like that. And so how do you handle that much data? This is a big problem in biomedical science in all sorts of ways, in all sorts of avenues. There are times when you can get the data, but it's so overwhelming, you don't know what to do with it. How do you analyze it? How do you figure out what's significant and what's just noise, right? And in this case, I want you to imagine that for each cell, we take it and we just keep track of 2 transcripts. Here are the 2 transcripts, and we could plot it on this XY, where for this particular cell, it was expressing this much of gene X and that much of gene Y. And this cell was expressing a lot of gene Y and a lot of gene X. And you can imagine that for only 2 genes, this would be a pretty efficient way to plot the data, right? Okay. Problem is, there are more than 2 genes. Well, what happens if we take 3 genes? Let's pretend there are 3 genes, X, Y, and Z, and we get a whole bunch of cells, and we plot each one of them in a 3-dimensional plot where how much they express gene Z is in the Z-axis, and how much they express gene X is in the X-axis, and how much they express gene Y is in the Y-axis. And okay, you can visualize that. You can see the pattern here, right? But it's already taxing us all, right? This is hard to tax it. But notice even here, the green cells all seem to be in this quadrant, and the red cells are mostly in that quadrant, and the blue cells are kind of scattered in different places. Oh, yeah, but you can visualize. Okay, now here's where I'll push your imagination. Now imagine we do a plot like this, and we do it in 20,000 axes. Shit. I know, it's a bit. 20,000 axes. Great. I can barely imagine 3 axes. You want me to imagine 20,000? Give it up. You're not gonna. But mathematically, the mathematics is there to deal with that many axes, right? Maria?
So if you're doing that stem cell, isn't all you come to like differential
At stem cell research?
What if you're going to be research using stem cells, let's say a level as well, for there is.
Hold on. That sounds a little more specialized. Let me hold on. But before we talk about particular kinds of cells, are you asking me, do stem cells express all the genes? Because the answer is no.
Can manipulate the right, it can differentiate the right cell, like.
Let's get there, but we are going to go after stem cells. Okay. All right. So imagine this plot, and you can't imagine it, but the mathematics is pretty clear. And now we're going to take that data and we're going to collapse it all into just 2 dimensions. Take all those points, each one representing one cell, how much it expresses each of the 20,000. We're going to, and there's an infinite number of ways you could collapse those together, right? Collapse them this way, this way, this way, and you get a different look for the 2-dimensional graph, depending which ones you did. But there are algorithms that try them all. It takes a long time for the computer to spit out the data, that tries them and ends up finding that collapsing that differentiates cells the most. So that you can do this and plot it on a UMAP. Let me show you where we're going. So that you can, and when you do this, here you get this 2-dimensional plot where we've collapsed all that data together. And look at this. All the tonsil cells are in this part of the graph, suggesting that they're all having a similar pattern of gene expression. As you'd expect, they're all tonsil cells, right? And the skin cells, interesting. They're up here in this part of the plot, and there seem to be at least two different subtypes of skin cells. Well, we don't know what made them different subtypes, but the fact that they have slightly different patterns of gene expression suggests maybe there's some difference in the kinds of skin cells there that we're not aware of yet. Or gut cells. So down here you can see, so umbilical cord blood, these are pretty close to your stem cells, Maria. Some of them are stem cells. Look at that. Their pattern of gene expression is in this part, and that part. This is known as a uniform map approximation projection plot, a UMAP. And that just says all that stuff that I told you before. And so there's an x-axis and a y-axis in terms of how they get plotted. But these axes, we don't have a name for what these axes are, right? They're just what worked to get the most distinction between all these cells in their pattern of gene expression, so that they indeed ended up in clumps together and distinct from one another. Good. Justin?
So when you're giving all this data, so Peter wants to know what the cell was before.
Yep.
A group we got.
That's right.
I could say, this.
In this case, you say, here's the results of tonsil cells, here's the results of the umbilical cord cells, etc. And now put them all together and make a UMAP. And what do you see? Cool. They're really cool. Because it's showing you differentiation. It's showing the differences between cells in terms of differentiation in the modern terms, which is to say, pattern gene expression. Maria?
He was because we talked about different factors that we can give to pluripotent stem cells to shape them into different types of cells.
Right.
This thought to validate what.
So maybe you could use a plot like this to confirm whether those skin cells you took, now that you've treated them, however, do their gene expression profile look like? Do they end up being in this portion? Yeah, that would suggest that they've got this right pattern of gene expression to be stem cells. But this class, yeah, Justin?
Are they weighting some expression of some beings more than others? So without these,
I don't know what that means. No, it is. This is how many transcripts we had of that gene, and this is how many transcripts we had of that gene, and etc. So, does the algorithm, when it puts the UMAP predict? To my knowledge, no. It's just
it's very different in this. You can catch yourself, you have how to say it, you don't get like that, and so you from it.
No, I don't think so. Although, of course, I could never. And I will say the algorithm, you have to put certain variables into it. But my understanding is it is of the infinite number of ways you could squeeze those points into 2 dimensions. This is the one that shows you the most separation. Evan.
So where we see lots of different cell types,
yep.
They're not kind of it's all start.
What? Oh, yeah. So I mean, and that means that there's places where the spleen cells and liver cells, they're kind of similar in terms of what genes they're expressing. Yeah, that's interesting too.
So different cells, but genes.
Yeah, because here they've color-coded it by which organ the cells came from.
All right.
But of course, we studied the nervous system. So people applied this UMAP to ask about whether, in fact, there's neurogenesis in adult monkeys. And they did it by taking an adult monkey hippocampus, dissociating all the cells, and then individually running RNA-Seq to make a transcriptome for each one of them. And they know, first they identify what kind of cells they are before they do the RNA-Seq. They do the UMAP, and here are GABA neurons. There are two different bunches of GABA neurons, and they have totally different gene expression patterns because they're in different parts of this plot. They all use GABA, but you might guess they're probably doing something different. Or here are some microglia. Look at this big hunk of microglia, and microglia have a very different pattern gene expression than all the other cells from the hippocampus. The important thing is that in the adult hippocampus, here are a bunch of cells that are expressing the genes for immature granule cells, which means they only recently came out of mitosis. And then you can go back to the animal and use other methods to confirm that, yes, here's some granule cells that are still expressing double cortin, which is only expressed just before and shortly after mitosis. And so in this paper, they were saying this is evidence that the adult monkey hippocampus still has dividing granule cells. And so we think the neurogenesis we validated by other means is probably real. Okay. If it makes you feel any better, one of the reviewers of the chapter said, that was really long.